Bismillahirrahmanirrahim, and welcome back to this next video. And this is the fourth video in the series of videos on the Alkaptonuria. In the last three videos, we generally discussed about the Alkaptonuria, that what is Alkaptonuria, why it is called is the black urine disease, how the problems in the metabolism of the tyrosine and the phenylalanine amino acid leads to the black urine disease, and we talk about the mutations that are responsible for causing the Alkaptonuria and those mutations that occur in the HGD genes. And the most common example of mutation that we discussed was the missense mutation, where one amino acid get replaced by another amino acid. We talked about the nonsense mutation, where their stop codon appear very early in the sequence of the messenger RNA, leading to a truncated non-functional kind of the protein. And we talked about the frame shift mutation, how the deletion in the sequence of the gene that leads to truncated non-functional abnormal protein. Now, in this particular video, I want to focus on some of the inheritance pattern of the Alkaptonuria. As we are talking about the genetic diseases, so we have to talk about the different inheritance pattern scenarios of the Alkaptonuria, that how the genetic makeup of the parents that can leads to different types of the offsprings for the Alkaptonuria. So we are going to start with the very simple one. We are going to represent the normal allele by the capital A for the Alkaptonuria, and the diseased allele by the small a. As we are talking about the human beings, we know that the human beings, they are diploid in nature. What I mean by that is that there are two copies. We are talking about the homozygous or the homologous chromosomes, not the homozygous, the homologous chromosomes. And on these chromosomes, you have got two copies for every gene. One copy of the gene that is coming from the mother, another copy of the gene that is coming from the father. So that can be the same or that can be different. So we are going to represent the normal allele by the capital A, the diseased allele by the small a. Say, for example, the mother's genotype, that is capital A, capital A. What I mean by that is that the mother is homozygous, but she is homozygous normal, is both copies of the allele, they are normal. If you talk about the father genotype, similar to the mother's genotype, he is homozygous normal. What I mean by that is that both copies of the alleles that the father is carrying, they are normal allele. So when you know about the genotypes, then we talk about usually the gametes. So if you look at the gametes, the mother can produce only the normal types of the gametes. I mean that all of the gametes of the mother that is going to carry the normal allele. Similarly, the father, all of the gametes that he makes, that will contain the normal copy, the normal allele for the gene of the Alkaptonuria. So when you make the Punnett square, here we are representing the gametes. These are the gametes of the mother. These are the gametes of the father. So the upper side and the left side of a Punnett square, that usually represent the gametes of the parents, and this right side of the Punnett square, that represent the fertilized product, or that represent the offspring. So if these are the gametes of the mother, so one of the gamete of the mother, that is going to get combined with the gamete of the father. And if you look at the genotype of this offspring, that is going to carry both normal copies of the alleles for the Alkaptonuria. Similarly, this one is the gametes, they are the same. So all of the boxes in the offspring, in the this particular part, they are going to represent the normal offspring. So if you look at the phenotypes of the offsprings that are produced by these parents, and the mother with this genotype, the father with this genotype, all of the offspring, they will be normal, and they will be homozygous in nature, and they will carry the normal alleles. Both copies of the allele, they would be normal. So this is the simplest scenario. Let us talk about the second scenario. Say, for example, the mother's genotype, she is heterozygous. So that would mean she is not normal. She is the carrier for the Alkaptonuria. And you need to understand the concept of the carrier. That carrier means that when you talk about the autosomal recessive disorders, the carrier would mean that he or she is not going to show the symptom of the disease, but he or she has the capacity to transfer the diseased allele to the next generation. As this small a, it is going to represent the diseased allele. So the mother genotype, she is heterozygous, and that would mean that she is going to be the carrier for the Alkaptonuria. And let us assume that the genotype of the father, he is homozygous normal, carrying both normal copies for the gene of the Alkaptonuria. Now, if you look at the gametes, the mother is going to make two types of the gametes. 50% of the gametes, they are going to carry the normal allele. 50% of the gamete that is going to carry the diseased allele. If you talk about the gametes of the father, he is going to produce all of the gametes which would carry the normal allele. Now, if you make the Punnett square, these are the gametes of the mother. So 50% they are having the capital A, 50% are having the small a. The father, all of the gametes, they are going to carry the normal allele. So if you combine this gamete from the mother with this gamete from the father, you are going to have a normal offspring. When you are going to combine this gamete from the mother to this gamete of the father, you are going to get a carrier. He or she is not going to show the symptom of the disease, but he or she is going to have the capacity to transfer this diseased allele to the next generation. Similarly, if you combine this gamete with this one, you are going to get the normal offspring. And if you combine this gamete with this one, you are going to get the carrier. So in this particular case, 50% of the offsprings, they are going to be homozygous normal, this capital A and the capital A, and 50% of the offsprings, they are going to be the carrier for the Alkaptonuria. So this could be the second inheritance pattern scenario for the Alkaptonuria. Let us talk about the third scenario. Say, for example, the mother, she is heterozygous. She is carrier for the Alkaptonuria with the genotype capital A and the small a. Again, the father is also heterozygous. He is the carrier for the disease. He is heterozygous, one normal allele and one diseased allele. Now, if you talk about the gametes of the mother, just like the previous one, she is going to make 50% of the gametes with the capital A allele. She is going to make 50% of the gametes with the small a allele. If you talk about the gametes of the father, similar to the mother, 50% of the gametes will carry the normal allele, 50% of the gametes that is going to show you the diseased allele. So none of the parents, they are showing the symptoms of the Alkaptonuria. Both of them, they are carriers. Now, if you look at the Punnett square, the gametes of the mother, these are the gametes of the father. So if this gamete combined with this one, a normal offspring. If this gamete combined with this one, you are going to have a carrier over here. If this gamete, if this gamete combined with this one, again you are going to have a carrier. But if this gamete from the mother combined with this gamete from the father, you are going to see an affected one. Now this is a very interesting scenario because both of the parents, they are not showing the disease. So when they see the physician and they say that there is an affected individual for the Alkaptonuria, so one of the explanation is that both the mother and the father, they are the carriers for the Alkaptonuria. And this scenario, when the gametes carrying the disease allele from the mother combined with the gamete carrying the disease allele from the father, that is going to give you an offspring that is affected for the Alkaptonuria. So 25% of the offspring, they are going to be homozygous normal. 50% of the offspring, this one and this one, they are going to be the carrier, and 25% of the offspring, they are going to get affected. So this one is an interesting scenario where both of the parents are normal, but you are getting an offspring who is a patient for the Alkaptonuria disease. If you talk about the fourth scenario, say for example, the mother genotype, she is homozygous, but she is affected. Both of the alleles, they are affected. The father genotype, he is affected. So this is a very you can see easy scenario. If both of the parents, they are affected, that would mean that the gametes made by the mother, all of the gametes are going to carry the affected the diseased allele. If you talk about the gametes of the father, all of the gametes, they are going to carry the diseased allele, and if you combine them, all of the offsprings, they are going to be affected for the alkaptonuria, and all of them are going to show the symptoms of the alkaptonuria. If you talk about the fifth scenario, for example, the mother, she is normal, she is homozygous, and she is normal. The father is affected, and both of the alleles the father is carrying, that is the diseased allele. If you talk about the gametes of the mother, she is going to make all of the gametes which carry the normal allele. If you talk about the gametes of the father, all of the gametes that is going to carry the diseased allele. Now, if you combine them, these are the gametes of the mother, normal one. These are the gametes of the father carrying the diseased allele. So if you combine this with this one, you are going to have a capital A and a small a carrier. A capital A with a small a carrier. This capital A with the small a carrier. This capital A with this small a is a carrier. Now this is very interesting. The father is showing the symptoms of the alkaptonuria, but as the mother, she is normal. She is transferring one normal allele, one copy of the allele that is normal, to all of the offspring. Thereby all of the offspring, they are going to be the carriers for the alkaptonuria. So the affected father, if he is going to marry a mother who is normal for the genes of the alkaptonuria, homozygous normal, so all of the offspring, they are not going to show the symptoms of the alkaptonuria. If you talk about the sixth scenario, say for example, the mother, again, she is homozygous normal. Now the father is heterozygous, he is not affected, he is a carrier. So if you talk about the gametes of the mother, just like the previous one, all of the gametes, they are going to carry the normal capital A allele. If you talk about the father, 50% of the gamete, they are going to carry the capital A, normal allele. 50% of the gamete that is going to carry the diseased allele. So if you make the Punnett square, if you can see over here, this gamete with this one, a homozygous normal. This gamete with this one, a homozygous normal. This gamete with this one, carrier one. This gamete with this one, you are going to have the carrier offsprings. So 50% of the offsprings, they are going to be the carrier, and 50% of the offspring, they are going to be the homozygous normal. So the genetic makeup of the parents, that does matter. So in some scenario, as I've told you, both of the parents, they are normal because they are the carrier, so they can give birth to an affected individual. And in one case that we discussed, that the father is affected, but because the mother, she is homozygous normal, so all of the offspring, they are carrier, and none of them is showing the symptoms of the alkaptonuria. So these was just some of the inheritance scenario when you talk about the alkaptonuria, and we will continue our discussion on the alkaptonuria in the next video.